So thank you very much for the opportunity to give you an overview of the A-WOL Drug Discovery and Development Program. And A-WOL stands for Anti-Wolbachia, and I'll introduce you to what Wolbachia is and take you through 2 5-year programs that have been funded by the Bill and Melinda Gates Foundation. So the neglected tropical diseases we're targeting are onchocerciasis and lymphatic filariasis, responsible for river blindness and elephantiasis. And these are some of these NTDs which rely on, in the case of onchocerciasis, just a single drug, ivermectin, which was just recently mentioned. And so this is obviously a high risk, and there is evidence that ivermectin is, or the parasite is developing resistance to ivermectin. So there's a real need for alternative treatments for both of these diseases. And this is, I think, a unique approach to treating a disease where instead of directly targeting the parasite itself, we are targeting an essential bacterial endosymbiont. So rather than using typical antihelminthic drugs against these parasites, we can now turn to the much larger field of antibiotics to see if any of these can be used successfully against the filarial nematodes. And here I've just summarized some of the essential biological processes that Wolbachia provides to the nematode. The first is that it's essential for larval development and embryo development. So areas where, or parts of the life cycle where you get rapid and extensive growth of the parasite depend on the presence of this bacterium. And you can see on the right there are some larval stages which we've removed Wolbachia from with antibiotics, and they're stunted and arrested. They will not develop into these large filarial adult worms. And these adult worms are a bit atypical for a nematode in that they live very long. So they can live for 10 or 14 years, whereas a normal adult nematode will live for a year or 2. And this longevity, again, appears to depend on the presence of the endosymbiont. And if we eliminate the endosymbiont, rather than living for 10 years, they'll die after a year or 2. So this gives us a very good macrofilaricideal activity, which is something which has been very difficult to develop against these diseases. Another thing that Wolbachia does is drive some of the inflammatory disease pathogenesis. So by getting rid of them, we can improve disease at the same time. But principally it's a macrofilaricidal, the ability to kill these adult worms. And nearly 10 years ago now or more, we have done clinical trials with a course, 4 to 6 week course of the antibiotic DOXYCYCLINE. This is very effective at getting rid of the Wolbachia. It sterilizes the adult worms, it blocks transmission, and importantly, it kills the adult parasites. And he does it with very good safety profile. Because, I'll mention in a minute, if you kill the adult worms or the microfilariae very rapidly, you can get some severe adverse events, which are a barrier to the expansion of some of the existing drugs in MDA programs. But the reason we can't use just this simple, cheap antibiotic in existing mass drug administration approaches is for filariasis, this is a relatively long course of treatment. Clearly not very long if you were comparing it to HIV or TB, but where we're used to a single dose delivered on an annual basis, then this is perceived to be a barrier to implementation. And of course, with this class of tetracycline, we cannot treat pregnant women or young children. So they're the real barriers to using that, and the aim of the A-WOL program is to overcome those barriers, essentially. And this just illustrates how it delivers a safe mode of action with this slow kill. Because here's an example where patients were treated some 20 years ago with suramin, which many of you know is a very toxic drug in its own right. But one of the problems of killing these adult worms rapidly is that you can get a reaction, an immune reaction to the death of the parasite, which can lead to these exploding nodules. Or in the case of lymphatic filariasis, where the parasites are in nests within the scrotal lymphatics, you can get scrotal inflammatory lesions. And I assure you, these sort of adverse events are not good for compliance. The other major safety issue is with another filaroparasite, which is on the right, called WOL. And this currently prevents ivermectin being used in much of Central Africa, because in this case, they have very high numbers of microfilari circulating in the blood. And if you kill those rapidly, as you can do with ivermectin, then you can get them blocking the capillaries of the brain and developing encephalopathy. So you get severe and fatal adverse reactions. And this is, as I say, a barrier to the expansion of ivermectin-based programs in Central Africa. So at the moment, using doxycycline, we can have a new macrofilarocidal therapy for both lymphatic filariasis and onchocerciasis. We've done regimen reduction experiments, and we get suitable efficacy with now a 4-week course of treatment in lymphatic filariasis. And importantly, we can halve the dose. So this is a dose which is typical for prophylaxis for travellers to avoid malaria, and this will be typical for the treatment of acne, for example. We can use it in areas where there's lower without any antihelminthic drugs. It's effective just delivered on its own. And in restricted areas, we can deliver this in a community-directed mass drug administration. So in areas, a population of about 20,000, we used the community to deliver doxycycline. We got a very good therapeutic coverage, and much to our surprise, 98% of the population that started the treatment finished it. And the reason for that was they felt good. Their skin started getting better, and they felt more energetic and better. So that's a very good way of ensuring adherence to long-term treatments. And what are the key benefits of having a macrofilaricide? You probably know that the programs for onchocerciasis and lymphatic filariasis are probably among the more successful of the NTD programs. But there are still some barriers to reaching the ambitious targets set by the World Health Organization in terms of elimination by 2020. So the programs are very long-term. With ivermectin against onchocerciasis, you have to give it annually or biannually for 20 years or more. So delivering this in these communities is a real challenge. As I said, we need an alternative treatment in the event of reduced efficacy or the development of resistance. We urgently need an alternative to treat patients in co-endemic lower areas to avoid this severe adverse event. We also need a tool in the end game, so where mass drug administration no longer becomes feasible or cost-effective, we need to have a test and treat strategy to treat hotspot areas and hard-to-reach communities. And I'll give you an example of that later. And of course, stocking the long tail of elimination. As you're all aware, there's this long tail of residual cases which a macrofilaricide would hopefully reduce the time frame. So here I've just listed the goals of the Anti-Wolbachia program. It's simply to find anti-Wolbachia treatments which are compatible with mass drug administration a shorter period of time, and we're currently looking at a regimen of 7 days or less, and a regimen that can be used in children and pregnancy. We have a secondary goal to find the best regimen with existing antibiotics which we can use in these more restricted settings where we have a problem such as drug resistance or the lower co-endemicity. This illustrates the main outputs of the first phase of our program. So we use existing drugs and refine the regimen to go down to 3 weeks with combinations of rifampicin and doxycycline. Most importantly, the dose was halved. We did a lot of screening of both registered drugs, focused libraries from pharma, and diversity library screening, which generated a number of registered lead candidates, some focused lead candidates, and some more diverse candidates which are progressing in our pipeline. We've collaborated with a large number of pharma partners who have provided focused drug libraries. So this is where we've given them a list of essential genes that we think are important to Wolbachia, and they've sent us compounds which they think are going to be effective against those targets. And we've also had a large diversity library screening activity, and most recently we've acquired the MMV box to screen against Wolbachia as well. The starting point of our screen is actually an insect cell line which is infected with Wolbachia. Here we use the operator and high content imaging system to use texture analysis to evaluate the elimination of the bacteria from the cell. Recently we've been transferring this assay to AstraZeneca to develop it into a real high throughput capacity supported by the WIPO initiative. We've had a number of challenges with our working with AstraZeneca. First of all, they decided they didn't want to work in infectious diseases, then decided they were going to move to Cambridge when they're just down the road in Macclesfield, and now someone thinks that they're going to take them over. So hopefully this development of this high throughput screen will happen in a rapid high throughput way, so we can use it before all this impacts on our activities. From the cell assay, we take it through our screening pipeline, so we go into a couple of, which is streamlined because this is one of the bottlenecks with long-lived filarial parasites. So we've streamlined it into a larval screen because this is very sensitive to anti-Wolbachia treatments, and then an adult Brucia malayi screen in mice. We are now running as a discovery and a development program, taking the outputs of our registered and repurposed screening and our more focused and diversity screening through to either optimize the best registered tetracycline, the best combination of anti-Wolbachia drugs, and we have a repurposed drug being developed with AbbVie. And we're undergoing lead optimization of now several hit series, working together with the Cheminformatics and Medicinal Chemistry team in the University of Liverpool, headed by Paul O'Neill, and in partnership with DNDi. So this is our current portfolio. So we have a drug which you can use, doxycycline, now in restricted settings. We have some point-of-care drugs which can improve upon doxycycline, but it's the lead series which we think are going to be most productive in terms of developing a drug to suit our target product profile. And more recently, on top of the Gates funding, we have also acquired funding from the GHIT Fund, which is an initiative between Japanese pharma and the Japanese government. We also use PK and PD modeling to inform our drug selection and development. And there's still a lot of innovation and new ideas that we can bring to the table. So we've been actually very successful at getting 3 of these Grand Challenge Exploration grants, where we are looking at turning the nematode's immune system against the symbiont by activating autophagy. So that gives us a cidal mode of action. We need a small animal model for onchocerciasis, and we'd like to develop a cell-based screen for direct macrofilaricides. I'll take you through some developments recently. So our final tertiary screen for onchocerca drugs is a parasite called Onchocerca roentgengi, but unfortunately that parasitizes cattle, and if you show this to any medicinal chemist, they sort of go white and sort of start getting worried about being able to supply that amount of drug for testing. So clearly that's a real bottleneck in our pipeline. So what we thought we'd do is try and take the parasites out of the cow and put them engraft them into some immunodeficient mice. And thankfully that's worked. So you can see some of the nodules here which develop neovascularization, so that they are viable and can survive in these mice for a long time. So we validated that model using both existing direct-acting macrofilaricides and anti-Wolbachia drugs. The other problem is we need a counter screen. Remember, lower is going to be a severe adverse event, a potential driver of severe adverse events if you have a rapid activity against the microfilary. The problem here is that lower models are even more challenging to work with. So we have mandrills and baboons, which are the only current models that we can use. And again, we've exploited immunodeficient mice, and we can take parasites and infuse them into these mice. So now we have a mouse model which we can screen against these microfilary. So as we stand today, this is we can use doxycycline as a macrofilaricidal under restricted settings, and we hope to have second generation and third generation drugs coming online to slot into the program as it moves towards the end game, where the need for a macrofilaricide will become more important. It's already been adopted, this approach, by the programs. So the onchocerciasis elimination program of the Americas is using doxycycline in some of their residual areas. So in the north of Venezuela, they're treating patients who still have persistent infection. And in the Amazon area, the Yanomami Indians, these are a nomadic tribe which wander around the Amazon jungle and are challenging to give ivermectin to every year. So they're being brought to health centres and being given a course of doxycycline to accelerate the ability to eliminate these and lead to certification of elimination in the Americas. It's also been adopted by the African Programme for Onchocerciasis Control last year at their Joint Action Forum. So here's a map of where you get lower in Central Africa, most of the forested regions, because it has a vector which lives in the canopy. And these are currently restricted from widespread ivermectin distribution. And so it's about identifying where we can deliver this alternative approach in these restricted areas. One of the added benefits of using doxycycline, which we hadn't previously considered, was it has an anti-disease activity. So in addition of getting rid of the adult worms, individuals that have clinical disease show improvements. So if you read all the textbooks, this is an irreversible, untreatable condition. So the textbooks are now wrong. And we can use this as a new morbidity management tool for patients suffering from elephantiasis in MDA systems. So I'd like to thank by acknowledging all of the team working in the AWOL program and many of our partners in Africa doing all the clinical trials, but also in Liverpool with Steve Ward, who's our pharmacologist and driving our discovery, and Paul O'Neill, who's responsible for the medicinal chemistry. And finally, thanks to the Bill and Melinda Gates Foundation for their vision in recognizing the need for a macrofilaricidal drug.